morning. Thank you for the invitation to come to Stanford. And those two talks were fantastic, right? You can see precision medicine shaping right before our eyes in our lifetimes, in our careers. Pencils are sharpening. And so what I want to do is infuse into that a precision form of proteomics and bring you to a level of data and a operational plateau that I've spent the better part of a career trying to bring into fruition. And it's coming, and the rate of that is accelerating. And so it's basically aligned with this simple idea, this simple slide, that the concept of a protein at the molecular level is not specific enough to capture our biology. And so whether you're using epitopes and antibodies or small peptide surrogates for what the world now calls proteoforms, these are creating another level of inference, more imprecision, more variance, more processes that were the genesis of my question of Mike earlier, which is like, are the data streams powerful enough to get the job done? Well, what is the job, right? If it's creating 1,000 associations and helping GWAS cross that divide of the genotype to phenotype gap, right? Because proteins are close to phenotype, right? Shouldn't that be true? Or closer? Are metabolites more valuable to connect to genotype than proteins? You know, that's a complex question, and we can't get a read on it exactly. And so lots of multiomics are vying for that vaulted position of what is the data type that is most correlative. If you're going to take 100 people and try and do associations, find out what's wrong, find out interventions. So all the decorations, and I'll come back to that theme in a bit. I'll give you a dosage of this proteoform biology. And yes, we're trying to digitize it like our famous uncles in genomics, that we seek to domesticate, regularize, and sequence the human proteome and digitize it and create all of that reality before 2035. So right today, the biggest delta in this is the technology, which I will give most of you a sense of this data type and a sense of the technological state of play, which of course has not had a $3.5 billion project like the Human Genome Project, which in this case is a human proteoform project. And we can come back to that in the Q&A. But what we have done is use single particle mass spectrometry to improve the sensitivity, drop the floor by 1,000-fold. This whole area, like a tree fell in the pandemic, and few people heard it. But we with our partners at Thermo Fisher, a scientific right down the road here, we were able to really change the nature of proteoform detection using mass spectrometry. We can now sniff proteoforms as they come flying off of human tissue at a reasonable scan rate, at least to a few thousand proteoform depths. We're using the exact same lawnmower, I'll liken it to a lawnmower, to do single cell proteomics at a 20-fold increased rate than other types of mass spectrometry platforms. And I'll try to get all through this in 28.6 minutes. So 17 companies is a lot. I'm rolling 5, but these 4 are the ones I'll declare for today, and 2 of them are actually profitable. So this proteoform term, it's not just a word, it's a movement. And that crescendo is happening. It started back in 2012, 2013, a group of about 50 like-minded folks with support from mass spectrometry vendors got together in Portugal and said, we need to lock down all these sources of variability in proteins, which are depicted on this slide. So on the far right is a specific proteoform, and it's depicted to have a combination of, say, a single phosphorylation with a single glycosylation state, which contains a coding polymorphism or mutation, and all the isoform variants is asserted as well. So you can see there's 2 alternative transcripts here. Uniprot calls those base sequences. And all that variation together is locked down with complete molecular specificity. That's what we're talking about here. Imagine, what if you could actually nail proteins in a compositional sense composition of matter, all the atoms you're asserting correctly on that protein molecule. How much would that close the genotype to phenotype gap? So this paper, which has been cited over 1,000 times now, continues to be our calling card. Yes, from genes to traits, this gap, this connector function. This is one of the promise of the proteoforms. If you could measure this at scale, which we cannot, and this is the purpose of trying to do it in use cases, turning to success cases. We've got dozens of those. And the dynamic in the field is that those sort of exist in a world that's distinct from the O-links and the SOMA scans and from bottom up. It's in this lane, and I'll bet many in the room haven't heard about what's the activity set in this swim lane before. But the value proposition is if you can make those measurements, you will find the isoforms and PTMs that are functional, that are valuable, that are dynamic and regulatory. And if that's true, we should be able to use them as deep connectors between phenotypes and come up with better biomarkers, durable to more pre-analytical variation, and all the sources of variation in healthcare research. And I'll argue at the very end that for drug companies, this could have a huge upside. The detection of the selection of drug targets. So for example, I was sitting next to a synuclein expert in Parkinson's, and we'll hear from her in just a few minutes, but imagine you could determine the synuclein proteoforms through the progression of Parkinson's disease. And if you knew that information, say a couple hundred proteoforms through, if you're talking about a few $1 million study design, what drug targets would you select? And when do these different isoforms fire? Does the protein get fragmented into pieces as in the progression of disease? Maybe proteases would be an interesting drug target, right? All of these things become possible with perfect molecular understanding at the protein compositional level. So moving along, I'm just tapping into 600 million years of evolution on the planet, right? PTM biology, we escaped the central dogma, and kinases and phosphatases arose a dance to create something different than gene regulation and post-translational regulation. This is something very simple. So I want you to think about that when you think about the MAP kinase cascade. I'll show you a MEK1 example. And I'm going to have to go quickly to show you these examples, but we're just tapping into some very basic biochemistry here. There's nothing new. And so let's do one. Let's do a quick example. We're going to do triose phosphate isomerase. It's the 5th enzyme committed step in glycolysis. It's the TIM barrel. What could you possibly learn more? There's 280 structures of this thing. And what are you going to learn new? Well, there's about a dozen or more PTMs that are known on the protein, but which ones are functional? So we drank our own Kool-Aid, did our thing with TPI, at least in the context of cancer cells in a dish. We found that serine 20, phosphorylation, was the dynamic PTM, present at about 20% stoichiometry. Long story short, that phosphorylation is adding to an already really fast enzyme. It makes it even faster by at least 400% or 4x faster, the catalysis rate of TPI when it has a single phosphorylation on the dimer. That's the story of this paper. And it was hard to synthesize the exact proteoform that we needed. And that was the limitation. And we couldn't get a structure of the monophosphorylated dimer. But and I'm having difficulty with the pointer. But basically down in the lower left is the structure of the TIM barrel. And that phosphorylation is sitting right here, right in the entrance into the active site. And so we hypothesized about why that phosphorylation could actually speed up a near diffusion limited enzyme. But that's an example of, okay, putting something through the proteoform process, finding a PTM that mattered, at least in that biological context. Okay, so moving forward, I'll just show you some, that was fun, let's do two more. Here are drug treatment examples just in cell lines. One's a histone deacetylase inhibitor, and you can see the increase in acetylated proteoforms on a histone H4. That was many moons ago. That is 2006. So what's changed in that time? Well, the technology I'll tell you about today allowed us, we can now go instead of something with huge copy number and small protein, but now we can go to something bigger, average size protein, and something present at a few thousand copies per cell, MEK1, in the BRAF, ERK, MEK pathway. And so bemarufinib is maybe known to some of you. It's a cancer drug used in certain mutation in melanoma. And what you're looking at there are the different phosphorylated proteoforms and the pattern that emerges through different models and through drug treatment. And you see the 1, 2, 3, 4 in relapsed melanoma models. You see a fourth phosphorylation. And there's 9 phosphorylations known on this protein. And so the proteoform landscape is nowhere near 2 to the 9 big. Every phospho could be on or off, right? So there's 9 sites. You go, oh, in the computer, there's 2 to the 9 phosphoproteoforms. But when a simple 2-second mass spectrum of the whole protein, you can see how many proteoforms are populated, what are their stoichiometry, and what's the dynamics in the system. So we are making progress, and there's a lot of examples. I think I'll just skip this for time, but there are now dozens of research groups. Many young professors have elected to dedicate their labs to focus on proteoform measurement, and we have one in the audience as well, Ruben Luo, here at Stanford, doing great work in translational top-down proteomics. And it's a very simple idea. It's like, if you have a molecule, don't shred it into 50 pieces and then get, a few percent, 10, 20% of the pieces back, which is a description of bottom-up proteomics, right? You would never do that in metabolomics. If you took a collection of a few thousand metabolites, started to disconnect the bonds between the atoms, and then get 10 or 20% of the atoms back, you wouldn't do it. But that's bottom-up proteomics, and that's all we've been saying is to have the value proposition be demonstrated enough that people start to think, well, we need to measure what we have to make progress, not what we can. And that conversion is an active discussion in modern proteomics. And there's many metaphors about how to describe this to you. This paper just has one figure, one page to try to explain at a better depth than I'm doing now what the difference is between bottom-up and top-down proteoform characterization. Right, and I won't even get into the inference problem. That's a very granular proteomics conversation. So what I want to do now is shift gears and just give you 2 small vignettes about the updated technology. We're excited about it. The last few years have come a long way. And I'm going to pivot in doing that by showing you, okay, so from composition, if you know the proteoforms that are present, how does that help you then go into space and go into single cell? So spatial proteomics, you've blown up huge, right? Antibody, multiplexed antibody-based imaging, the HubMap group that Mike helps lead is just, doing amazing work in spatial and single cell biology. So of course, we're trying to catch up with that train, but at the proteoform level. And so let me show you how that's done, at least in our lab to date, and what the nature of the tree that fell in the woods in the pandemic, what it is, just to give you something to take away from the talk. So because of HubMap, we got into this world and we came into conversation with Julia Laskin, who had years ago just invented this thing. It's 2 fused silica capillaries you can see in the left of that slide. Those 2 capillaries come together, there's a little droplet there. That droplet, think of it about 100 micron in size. And that little droplet is doing a micro extraction, and there's liquid flowing out of the capillary into the entrance of the mass spectrometer. And that is nanodesi or desorption electrospray ionization. And you move the droplet. About 40 micron a second, you're moving the droplet up and down a tissue slice, a thin sectioned piece of cancer tissue or healthy tissue. Okay, so that was great for lipidomics and lipid imaging, mass spec imaging, huge thing. Wasn't great for top-down proteomics, or when we didn't have the sensitivity. Now we do. It's 1,000-fold improvement in the concentration sensitivity of the method, and we call it I squared MS. We've got about a dozen papers on this. And this allowed us to be able to, if you'll allow the lawnmower analogy, we're mowing, that droplets are a little blade that's cutting the grass. And what's coming out, the blades of grass are proteoform ions. They're proteoforms that they're being micro-extracted and thrown into the mass spectrometer. And because we didn't have the sensitivity until recently, this wasn't a thing. But now we can go up to 80 kilodaltons, 100 kilodaltons now even, and we don't get single cell resolution. This is 40 or 50 micron. Just set it up and do it. It's about that, spatial. But we can get into cellular neighborhoods. And let me just tell you a little bit how the lawnmower works. I know maybe this isn't the optimal crowd for that, so I'll try and go light on you. But this is the fundamentals of the mass spec. Old school at the top, new school at the bottom. The first thing we had to do is dilute the sample or get 1,000-fold less ions into the instrument to get down to the single ion regime, which is what you see in the lower left. That's 50 ions. This is of a 280 kilodalton protein complex. And so there's 50 ions sitting there. The trick was to move from a situation where you were using the constructive interference of all the frequencies of the ions inside a mass spec, an ion trap, an Orbi trap. The constructive interference was the genesis of signal. And that's what was changed. Each individual ion is the genesis of its signal. So you no longer need constructive interference. Each signal is tracked over about a second observation period. And that slope is the charge of the ion. And once you know charge, you can back out the mass of whatever that ion was. And that's what allowed a big improvement in not just resolving power, but a 1,000-fold concentration, sensitivity increased. And no matter how complex the sample, you're still going to get a mass out of it. And this is a depiction of it.
Individual ion measurement.
You'll just see 4 ions moving along, and the different slopes you see in the bottom right. The frequencies are really stable, 1 part in 10 to the 8. So the mass precision is incredible. The problem was the charge. You had to determine how many charges. Was there 38 charges, 40 charges on the ion, etc. Okay, so there's your deep dive into mass spec. Coming back out for air. Here's just it in action. This is from a single woman with serious ovarian grade cancer, and there's tumor, standard IHC here on the upper left. Bottom line here, long story short, you've got proteoform biology encoding spatial position. We have a few examples of this. This is a small protein CRIP1 depicted in the lower right. Comes in 4 different proteoforms. The ones I'll focus on are just the unmethylated arginine there in the lower right, monomethylated arginine, and dimethylated arginine. I'll just focus on the extremes. When you're unmethylated, you are really focused on smooth muscle cells. And so what you're seeing here in this, if you can follow my pointer, that is a vasculature feature, and it's coming in and out of the plane of the board. It's sort of depicted in the upper left here. You can sort of see the vasculature pointed out. That's what that is. And we've seen this now across different organs, that this protein CRIP1, when it's unmethylated, it hits smooth muscle cells, whereas dimethylated form really is localizing it more to the tumor. Suggesting, I wonder what the arginine methyltransferase is that does this. And might it not be an anti-cancer target? Okay, so moving right along here, we're going to use the same lawnmower, and we're just now going to mow over single cells. That's basically it. So I hope you can take away from the talk something permanent in your mind, and let's look at how the lawnmower does its thing. So here's the same setup that I showed you, the capillaries there in the upper left. The droplet's going to be even bigger this time, maybe 150 micron in size. But all we're going to do is create from a single cell suspension, we're going to drop cast a relatively dilute concentration of single cells onto standard ITO slides. And when we do that, we're going to create a large cell-to-cell distance so that droplet can just start mowing over single cells. And you can probably imagine what's going to happen when a droplet comes mowing over a single cell. And so I think I got a video. Yeah, when it comes over it, here we go. What happens is you get a burst of proteoform ions. And the bigger the cell, the more the burst. We've seen up to 50,000 ions at a time. And we now need to know how to collect all of those blades of grass. You can sort of see a burst there. Um, uh, and that's a single peak from a single cell, which we've developed software to characterize and capture. And yeah, here's 20,000 ions. This is from a population of NIH 3T3 cells, so it's a clonal population of cells, and that's what came out in March in that paper there referenced. But each little burst is a peak, right? And if you line up this for 9, 10 hours, like in the lower left here, we call this now a cellogram because these peaks are like a chromatogram. And that cellogram, we can filter it with our software. And so there's 600 single cells, and you can see the throughput increase for mass spectrometry-based single cell proteomics is immense. And the big limitation is the cell-to-cell distance. But if we just take through all the data, all the ions, we can aggregate them, and that's what you see in the lower right is the mass spectrum, and then we can identify those proteoforms, in part because we already know the proteoforms, at least 10,000 proteoforms deep from NIH 3T3 cells. We have a reference library of proteoforms that helps us assign that's histone H1.4 with an N-terminal acetylation and a single phosphorylation. So now we're not going to not do cells in a dish. We're going to take rat hippocampus, create a single cell suspension, and drop cast. Pardon me, especially for those online. Okay, and we're going to do the same thing. There's the peak. There's the droplet in the upper right, hitting the cell. And mostly, as you know, probably out of the hippocampus of a rat, you're going to get neurons and you're going to get astrocytes primarily. And so those two cell types are depicted, and you can see where this story is going to land, which is we now need to take the proteoform signals that we get out of these single cell events and actually categorize and end in the same place that single cell RNA-seq lands. Okay, and so here's every couple months we get a new prep and we get more proteoforms out per cell. We have a collector's curve for the proteoforms. Now we can get over 500 identified proteoforms, and that's from a 5,000 cell run. So we're running around, between 500 and approaching 1,000 cells per day. And so now here's the clustering. Here's where I told you where this is going to go. So using gene set enrichment, Indira, a great biostatistician in the lab here, originally from Cuba. She's doing some great stuff, and she just ate this for lunch and turned this around that, hey, yeah, here's your astrocytes, and they are metabolically active. So they do a lot of glycolysis and other primary metabolism is what's driving that clustering of astrocytes. And getting closer to the UMAP, or I'm not even showing you a TISNE, which looks a little bit better, but just standard PCA for now. We've got the ability to separate astrocytes from neurons and even some microglia there in blue. And enzymes like aldolase are driving this for separation. And in astrocytes, the lights aren't super low, but you know, aldolase is a really good marker of astrocytes along with GFAP. And so those co-stain, as you can see there in a published result. So this is coming along nicely. Of course, we'd like to get more separation. We'd like to get it looking like single cell RNA-seq. Of course, that's our aspiration. But I think this might be the mass spec platform that gets to 1 million cells first in terms of the data. Now, the depth of the data processing, we're scurrying as fast as we can to get all that set up. And now I hope you can see not just what proteoforms are, but how the compositional knowledge and a library of them can help you move into space and into single cell. And we're operating at this single ion limit. The thing in the lower right, warning, okay, here comes another dose of mass spectrometry for you. Most of you not used to looking at stuff like that in the lower right. But the library proteoform is what's in the butterfly plot pointing downward. Those are the isotopes. Those are the carbon-13 isotopes of an intact protein. And that's our fundamental unit of currency is a proteoform. That's what it looks like in a computer. And you can see now down to this single ion limit, right? We're statistically limited in what we can collect from a single cell. Those are actual data sets from 2 different proteins. So we've got some nice FDR controlled new score that we're all very proud of, but I'll skip telling you about today. And it's published, there's a bioarchive out there for a proteoform score. So we can convert our individual unit of measurement into a proteoform assignment score. And that's part of a general thing, I think, across, well, ideally, the human body and biology and all the tissues and organs and cell types that we could bring this kind of clarity to the proteome. I mentioned single cell RNA-seq. You look at that growth rate, right, from the beginnings of all of this with the Human Cell Atlas and with HubMap and a lot of the early work with Steve Quake and Aviv Regev and Sarah Teichmann. I mean, look at what has happened in there. The 1 million cell threshold was hit maybe 2016, 2017. And then the growth behind it, reduction in cost, economies of scale, private sector impact. This is what I'd like to see and what I'm agitating for to bring to the world of proteoform biology. It can be digitized, it can be measured, and the value prop is really the centerpiece because that's for 20 years what I've had a problem articulating it in a way people were ready to hear, or maybe it's how I say it. But I think that value proposition, this knowledge, will it be as transformative as what Jessica started off her talk with, the Genome Project being transformative, changing fundamentally biological research? Could it be that impactful? I'm betting the majority of a career on it. So I'm going to keep saying those sort of things. And I just invite you to think about, well, why do I have to learn the proteoforms to then find out the isoforms and PTMs that matter? Is that really the path that will clarify protein level biology? I think it's part of the solution, but why would I even say that, right? And I think a lot of people think, well, the antibody-based platforms, Western blots, antibody-based imaging, peptide-based mass spectrometry, that'll get us there. And I respectfully disagree with that. So with that, I hope I've given you some perspective and that you take something away from the talk. I really appreciate the invitation. I do think it'll be a major step forward, and this idea of being proximal to phenotype and closing this genotype to phenotype gap. The proteoform as a connector, right? The association between molecular entity and phenotype. We have initial examples where proteoforms have served that function, that connecting function. And we need to do a proteoform-level association study. And I think the technology is getting to the point where that can be done. It's just not widespread in a lot of labs. So with that, I'll close out and hopefully we can have questions and vibrant discussion. Those are the people who help pay the bills back in Chicago. And thank you for the invitation.